It's a pleasure to introduce Steve Evedon from Ohio State University. He's going to give us a talk today with a really great title called Phage Therapy, Pharmacology, Ecology, and Spatial Structure. Really looking forward to hearing this, and maybe seeing some skateboarders that he was from the city.
No skateboarding videos, unfortunately. So this is kind of an amalgam talk, taking together 3 talks that I've given over the past 6 months, 3 very different talks. So it's going to cover a lot of ground, and I'm hoping that it'll be, at least for a little while, kind of my standard talk, but this is my first time giving it. We're under time constraints, so who knows. So it is going to cover a large amount of things, and well, first of all, so I feel compelled to point out to most audiences that is not Ohio. Instead, it's Sedona, Arizona, and I spend about a quarter of my time there. And this is where a lot of the thinking that went into at least some of the stuff here was actually done. And I was giving a talk in China in front of an audience who had apparently absolutely no idea who I was, and I was told I should somehow introduce myself, and the most appropriate way that came to me for doing so was to just put my books up there. So I've unfortunately been authoring and editing, mostly editing, way too many books. And I've got 3 more that are in progress. But one of them is supposedly kind of done, so I'm very happy about that. And I've come to the conclusion that I don't want to edit any more books. I'll author books if somebody should decide that I'm such a bigwig that they kind of want to have me as the senior editor that does nothing. I'll consider that, but no more books. But anyway, so this is based upon 3 talks, all kind of smushed together, and what I am doing is going to use those talks as kind of like the organizing principles. And the talks are, one of them was Considering Phage Therapy, Pharmacology, the Pulmonary Perspective, and that was actually in a town called Yanji, China. That was in August. And another one is the Applied Ecology of Phage-Mediated Biocontrol that I gave in September in Hiroshima. And then I gave one, Ecology of Phage Biofilm Interactions, Theory and Application, which I gave in Zurich. And actually, most of that one's going to come at the end. And that has kind of the most sciencey stuff in it. The common theme, however, is going to be ecology, it's going to be spatial structure, and it's going to be phage therapy, and I'll just keep going back and forth between these things. So this is the organizing principle. So what I've done is I've taken these titles and I've underlined the part that I'm about to talk about. So just a brief introduction to bacteriophages. So phages are the viruses of bacteria, and they actually come in a fairly substantial amount of forms, morphological types, although not nearly as great amount of diversity as seems to be associated with archaeal viruses. The viruses of archaea. The ones most people are most familiar with are the tailed phages, but there are also phages that have lipid associated with them and don't seem to have tails. And then there are the smaller phages. These are relatively big genomed with tails, double-stranded DNA. These are also double-stranded but medium-sized genomes, and they have lipid associated with them and no tails. And then these are the single-stranded, both single-stranded DNA and single-stranded RNA phages. And this is what I tend to think about. For me, these are the other things. I have a strange need to make lists. I gave a talk some years back at the Pasteur Institute, and I was eating lunch with grad students, and one of the grad students said he was looking at my stuff online, and he came to the conclusion that I like to make a lot of lists. And indeed I do. And this is just lots of different things that phages do in the course of infections. And what I tried to do is take every single concept that I could think of and put it into a single figure, and attempt perhaps to make the figure as complicated as possible. And I actually put it into the book, the one sole author, single author monograph that I published. If only for the sake of having something that complicated in there. So there's no need to really consider it in any detail, but that's of course the beetles, the yellow submarine, And that's just circling a lytic infection. So a lytic infection, you have a phage absorbing to a bacterium, you go through some kind of infection process, gives rise to the synthesis, maturation of phage particles, and ultimately the release of those phage particles, such as veolysis. And I put this in there, just that having it fade away as a pause to remind myself. So what I do is I do a lot of different things, but one of them is to think about different aspects of phage life cycles that are kind of interesting. And then I try to explore them in some kind of detail. And this has a historical component to it. So one of the things that I've just recently done and just recently submitted is the idea of phage secondary infection. And on this figure here, there's actually 3 places where we have secondary infection, but it turns out that that's all secondary infection of a single specific kind, and there's more than one type of secondary infection. So it's useful going out into the literature to try to figure out what people are talking about. And very often you find that different people are using the same words to talk about completely different phenomena. And secondary infection is one of those things. Secondary infection comes from medical microbiology and epidemiology, and has been co-opted within the phage literature, meaning approximately the same thing. I first understood it as basically being the equivalent of superinfection, where you have one phage that infects an already phage-infected cell. But it also has a component that comes from epidemiology, where you have a phage infection that gives rise to new phages that go on to infect new cells, and those are also described as secondary infections. And it's my belief that people who understand secondary infection in these terms don't really understand it in those terms, and vice versa, thereby leading to confusion. And then there's yet another concept that actually has some phage applicability as well, although it's kind of forced. So I do things like this. And I particularly like it when it's some kind of term that I can do massive searches on multiple PDFs, where I can find every single paper in my collection that actually talks about it, and then I can generate some kind of better understanding of things. So these are papers that talk about general qualities of phages and ecology, general ecology of phages. And also, I've done a little bit of writing on the role of phages in bacterial pathogenesis, and then there's the big one, phage history. And I put these together for my Zurich talk because I decided that people needed to know that I was actually publishing perhaps more than they understood I was. And people came up to me and said, wow, that's a lot of papers. So apparently they didn't know I was publishing as much as apparently I am. So there'll be a couple more of those. So moving on to phage therapy. So phage therapy is the use of bacterial viruses to treat bacterial infections. Although people use phages to eliminate nuisance bacteria in a larger variety of circumstances. And so a broader definition is simply using phages as a kind of antibacterial biocontrol. There's actually been a continuous 80-year history, or 80-plus-year history, of clinical use of phages. And this is particularly in the Republic of Georgia, Tbilisi, Georgia, in the former Soviet Union, where they've been using phages as anti-infection agents for a very long time and have lots of expertise. Phages are natural products. You, of course, can isolate them from environments such as sewage. They're abundant and diverse, and diversely bactericidal, probably more so than antibiotics, which makes them very useful as antibacterial agents, because it's much easier to isolate a novel phage than a novel antibiotic. Antibiotic resistance doesn't tend to give rise to resistance to phages, so if you have an antibiotic-resistant infection, then you can use phages to treat that infection without really any concern that the bacteria are going to be, as a consequence of their antibiotic resistance, resistant to phages. Very importantly, they show a low toxicity. I've been walking through this pulmonary infection treatment with phages literature just recently, and one paper after another, admittedly very old papers, but one paper after another says we added the phages did a good job, and there really weren't any side effects. So there's this low toxicity that actually contrasts to some degree from what you see with a lot of antibiotics, where you start getting into new antibiotics, you have to start worrying about the toxicity of the antibiotics. Phages can also amplify their numbers in situ, which is much different from what antibiotics do, but it's not essential that they do that. They can display favorable shelf stability, so even though they're viruses, they can be formulated in such a way that they're not going to just become inactivated in very short periods of time. So they actually have some potential to serve as medicinals. And there's also a lot of phage companies that are doing development of phage therapy. And this is something that I've been putting together for a chapter. It's not perfect. I have to actually go back into my database to figure out what's out there. But this is just a list of phage therapy papers, and you can see that there seems to be an increase, kind of steady increase in the amount of publications on phage therapy. There's a lot of review articles as well, but that's not included in there. I like making lists. So I was giving a talk at Hiroshima University at a symposium or seminar or whatever they want to call it on the use of phages to treat bacterial infections of plants, which people do. And so I decided to try to come up with the entire literature of phage used to treat bacterial infections of plants, and this is it. So there's a lot of people that are doing a lot of things with phages as antibacterial agents. I've begun blogging. Arguably, I've been blogging for a long time, but now I'm formally blogging. And so what I do is I do things like generate that list of antibacterial infections of plants, and then I just turn them into blogging posts. I actually wrote a blog while I was here. And this particular blog is the blog associated with the ISBM, International Society for Viruses and Microorganisms, which I'm responsible for the website. And on that website, there's a list of companies that are primarily involved in development of phage-based technologies. A lot of these are phage therapy companies. And I also have a website which I've been more or less neglecting for a while on phage therapy. We have a lot of papers listed and resources, whatever. It's phagetherapy.org. It's also phage.org, which I've been neglecting for even longer than that, and that's mine as well. So the pulmonary perspective, so I was asked to give a talk in Yanji, China. It was the first talk of the conference, although I didn't really know that a lot before I kind of arrived there. But nonetheless, it was the keynote talk, and they told me that they wanted me to give a talk that basically wasn't on my own research. So I had to do it on something, and I'd been delving into this literature, and so I gave it on that. And this is kind of a summary of what I've come up with, and I've packaged this up, and actually I submitted it on Sunday. So I submitted it from a hotel room in Vienna. These are treatment of pulmonary infections through the years, 1936 to 2011. And just going over here to the efficacy, you can see that they seem to have a fair amount of efficacy in treating bacterial infections with phages. I mean, these guys know what they're doing, and phages seem to be, if you sort of know what you're doing, effective at treating bacterial infections. And this seems to be, in fact, more so with treating of humans than it is in animal models, because animal models, there's problems with actually putting together the model of disease. And there's also problems in how people try to approach the treatment of infections once they've put together the models. But with humans, they basically take the phages and they apply them in some manner, multiple different manners, to the infections, and they just keep applying them until, for weeks or even months, until they've substantially reduced the bacterial infection. They basically treat bacterial infections as a problem, especially chronic ones, that need to be gotten rid of, and they hit these things hard, and they seem to have success. This is the Poles in Wrocław, Poland. There is a group that's been doing phage therapy for decades now, and the darker colors is the greater amount of success, and the cutoff point is basically right here, where they've had a fair amount of treatment success below that, and above it that indicates the less success. So we're talking, for this group, in the range of about 80 to 90% success rate. But the thing to keep in mind is that what they're treating here, by and large, are chronic antibiotic resistance infections. These are patients that are sick, have been sick for a long time. They've tried to treat and cure these individuals using standard approaches. They've been unsuccessful, and it's only then that they try phages. And they tend to have a fair amount of success in their treatment. So this is not taking the easiest cases and still getting 80% or whatever success. And then this is actually an animal model that I'm quite impressed with. So these were mice, and this is a Burkholderia cystic fibrosis model. And what they did is they immunocompromised the mice before they supplied the bacteria via an aerosol. And they then waited 24 hours, which in the animal model world of infections is a long, long time. A lot of times these infections either kill the animals quickly or they don't really become established for all that long. So waiting 24 hours for the infection to become established is actually rather impressive. And they followed this up with an aerosolized treatment dosage of the phages, which is also not easy because it's not easy to get an animal to, in fact, breathe in an aerosol. And when they did this, what they got after a 3-day period without phage treatment, this was how many bacteria were in the lungs, and with phage treatment, they have got about a 4-order of magnitude drop in the amount of bacteria, which is impressive. There's another study back here where they were treating Staphylococcus and Pseudomonas in a cystic fibrosis patient. And although they didn't get rid of the bacteria, they were able to reduce the amount of antibiotics they had to give this patient by about 50% following this intense, in fact, phage treatment. These guys, there we go, these guys probably could have done better if they didn't limit themselves to just a single phage dose. But they did that because of problems with maintaining the immunocompromised state. So they really didn't have all that much time over which they could do the treatment. So I found this to be very impressive. This is Semler et al. 2014. Very impressive phage therapy group. So phage therapy pharmacology, this is what I tend to do. So phages are a subset of antimicrobials. Like antibiotics, they are selectively toxic agents. In fact, highly selectively toxic because they can be very toxic to their bacterial targets and not toxic at all to the patient. And you can apply phages in a number of different ways, just like you can apply drugs, topical and parenteral into the blood. And supposedly people have had success also with Peros, where you actually have phages taken orally, and they can be taken up in some capacity into systemic circulation and find their way to infections and actually replicate once they reach the infections and at least contribute to the clearance of infections. The topical treatment is something where you're capable of applying large amounts of phages to an infection, potentially overwhelming the infection without having replication. Whereas parenterally, you probably do need replication once the phages reach the target bacteria. And then you can talk about these standard pharmacokinetics where you add the drug, or the phages in this case, and you have movement of the phages into the blood and movement of the phages out of the blood to body tissues, and you thereby get some kind of peak density in the vicinity of the target bacteria. And then without further dosing, you get a decline in the amount of bacteria that's a consequence of inactivation, sorry, not a phage, that's a consequence of inactivation or potentially excretion from the body. The immune system can get rid of these things. And the trick with any drug is to maintain sufficient densities that you're above the minimum effective concentration, but you're below the minimum toxic concentration, and therefore you can, over time, reduce the symptoms associated with whatever disease you're treating. So with phages, one of the advantages is that the minimum toxic concentration is rather high. So it's easy, seemingly, to have a phage dose that does not result in toxicity. And this is really important because of the other unusual thing about phages, and that is that they're actually able to increase their numbers once they reach the infections they're treating. So you, in fact, don't have a whole lot of control over the amount of phages that are present in the body, because it's not just dependent upon how many phages you add. So the second thing here, which is unusual for a quote-unquote drug, is that you have an ability of phages to go through what can be described as metabolism in a pharmacokinetic sense, thereby increasing their numbers in tissue in association with the target bacteria that they're actually killing, and thereby reach the minimum effective or higher concentrations, but without having toxic concentrations. Of course, this is important to choose the right phage. There are phages out there that carry toxin genes. You have to make sure that you're not using those phages when you're treating patients. And you can use bioinformatic means and others to make sure you're not using such phages. So there are two basic types, although as I'll get to at the end, in fact, more than two basic types of ways that you can treat phages. You can either, bacteria with phages, you can either supply so many phages that they overwhelm the bacteria and kill all the bacteria, and that can be described as inundative or inundation therapy or treatment, or passive treatment it's also called. Or you can have the use of phages that then have to replicate to reach sufficient high levels to get rid of the target bacteria and actually get replication in situ. If you're going to have inundative therapy, you've got to be able to get lots of phages to the bacteria, and that's going to require usually local treatment, topical treatment, where you can actually add the bacteria directly to the phages that you're targeting. Whereas this active treatment, where you have to have phage replication, you can supply phages to locations that are less accessible to the individual that's supplying the phages. But the problem here is that this active treatment requires performance characteristics on the part of the phages that are much greater than you see if you're just supplying phages directly. If you're supplying phages directly, you need an absorption and you need to kill the bacteria. If you're supplying phages in a way that they actually have to increase their numbers, then you have to not only have phages absorbing to bacteria, but they also have to go through replication, release new phages, and overwhelm the target bacteria that way. And although it's possible to come up with the phages that have sufficient performance characteristics, that's still not enough, because key also is that you have to have enough metabolizing bacteria to raise the phage concentration sufficiently high that you have essentially overwhelming or inundative densities to get rid of the bacteria. So although this allows you to get away with getting phages to locations where you otherwise would have difficulty supplying them directly in high numbers, you're still highly dependent upon what the infection looks like. And there are, as a consequence, great limitations in what you can treat using this active therapy. This I just came up with a kind of, just looking at the different kinds of therapies that are appropriate under different conditions. And what this has down here is just the amount of phages that you actually have to add. And also the densities of target bacteria. If you've got enough bacteria around, you can get away with active treatment. And in fact, if you've got enough bacteria around and you should happen to have the right phage find its way to the right place, you may not even have to treat at all, because endogenous phages may be able to, in fact, replicate to sufficient numbers to keep a bacterial population under control. But if you have lower bacterial concentrations, then you have to use passive treatment or inundative treatment. And somewhere in between is this idea of passive active treatment, where you actually supply a lot of phages, and there might be enough phages to get rid of the bacterial population you're targeting, but the phages may also replicate in the course of this and increase their numbers even further in the presence of their target bacteria, thereby even better reaching a point of overwhelming the target bacteria. And this, in fact, may be a kind of idealized way in which phage therapy can work, where you're not only supplying as many phages as you possibly can, but also allowing the phages to replicate to even higher numbers and thereby eliminate bacterial populations. What I was trying to do, though, with this is draw distinctions between the ecology of phages and the applied ecology of phage therapy. And one observation is that there's this idea of kill the winner, where in natural populations, particularly in aquatic environments, if you have a high enough density of target bacteria, that might be able to support phage growth to even higher densities, to the point where the target bacteria, the winners, are driven to much lower densities. And in a sense, this is equivalent to what you're attempting to accomplish with active treatment, where you're counting on the phages to replicate to higher numbers. You really need winner densities of bacteria in whatever population you're targeting. And that's not always the case. And so a difference between the applied ecology of phage therapy and the ecology of phages is that you might want to apply much higher densities of phages that normally would be generated within natural environments. And there's also a phage inundation threshold. It's a calculation of the number of phages that are required to, in fact, get rid of, without phage replication, a bacterial population. I'll get back to that. So these are just my phage therapy and my phage therapy pharmacology references. So nobody knows I publish anything, so I felt compelled to tell them. So ecology of phages. So this is just going through this kill the winner, and actually, because I'm into history, and I know that that's not a big deal for evolutionary biologists, but it is a big deal for the molecular types. There's a lot of different predecessors to the idea of kill the winner. But actually, I have this up here because I decided that what are the most basic things that are important to the ecology and understanding the ecology of phages. And the first one is the density of bacteria that you're targeting. And this determines how fast the viruses, the phages, can find the bacteria. It determines the phage generation time, because the longer they have to spend searching for a new bacteria to infect, the longer their generation time. And it also determines what can be described as the effective burst size. The longer you have to spend to find a new bacteria to infect, the more likely you're going to be inactivated. So the total number of phages that actually are effective in finding new bacteria to infect is lower than the number that will come out of an infected bacterial cell itself. It's also an important determinant, as you would expect, of the rates of phage population growth. And in terms of phage therapy, it's a very important determinant of whether or not you can get away with this active treatment, use high densities, or instead have to use passive treatment, where you have insufficient phage numbers to support insufficient bacterial numbers to support substantial phage population growth. There's also something that I'll get back to that I call active penetration, which is actually the movement of phages into spatially structured clumps of bacteria, where that movement can be aided by the phage processes, particularly of lysis of the bacterial cells. So second thing is phage density. So there's bacterial density, then there's phage density as important determinants of phage ecology. And so phage density is important in part because there's a transition that's made between the phage life cycles, life history characteristics, when you're going from there not being a whole lot of phages to there being lots of phages. A notable example of that is that lysogeny, where the phages form a latent infection, is much more likely when the ratio of phages to bacteria is higher than when it's lower. In terms of applied ecology, statistically, in order to kill off lots of bacteria, you're going to have lots of phages. And in fact, those phages are going to be absorbing with a Poisson distribution. So you're going to have a large ratio of absorbed phages to bacteria in order to kill off bacteria to a substantial extent. And generally speaking, you add, in phage therapy, you add your phages, you potentially have phage amplification, you have losses due to dilution of the phages, and you have losses due to the inactivation of the phages. And together those will determine your phage density, and if you don't have a sufficient phage density, you're not going to successfully kill off your target bacteria. And then this is something that people implicitly think about, but I don't think less so explicitly. You not only have to have lots of bacteria and lots of phages, but the phages have to be effective when they interact with the bacteria. And what we have here are lots of mechanisms by which bacteria display a resistance to phages. And you can think of these mechanisms as mechanisms that have the effect of degrading phage performance. And overall, you can talk about the phage absorption rate, how fast the phages are able to attach to and infect bacteria, which can decline as a consequence of bacterial characteristics or phage characteristics, decline in the amount of phages produced per infection or the burst size, or actual increases in the phage latent period, giving rise to longer generation times of phages. So these are degradations in phage performance, which are obviously of concern if you have phages that you're trying to use to kill off bacterial populations, but also this is an important consideration of how fast phages are going to be able to replicate in the wild. And then you have phage penetration, which I don't think anybody thinks about. Well, not quite, nobody. And this is the ability of phages to actually reach the bacteria that they're targeting. And this is an issue when bacteria form into biofilms. And the question is, how do phages get into those biofilms to, in fact, infect the bacteria? And then serially infect the bacteria. In fact, go from one infection to 2 to 3 in a biofilm. And then, of course, with those 4 characteristics, the bacterial density, the phage density, the phage performance, and the ability of the phages to penetrate to the bacteria, we can add a whole lot of more things. And this is just another example of where I was asked to do something. I decided to sit down and come up with as many interactions as I could think of that were phage ecological. And we'll get back to that. And then these are just more papers. So the applied ecology of phage-mediated biocontrol. So getting back to that, so this is just that list again. I decided it needed to be updated and kind of described more in terms of interactions and thus ecological aspects of phages and bacteria. So I made a couple of changes to this. And then I decided to go through it and try to figure out which one of these things is relevant to phage therapy, but not necessarily, which one of these things is relevant to phage ecology, but less relevant to the ecology of phage therapy. And so I kind of grayed out the parts that are relevant to phage ecology, but not phage therapy. And then I still decided that this was way too much to even bother going over when I first generated this, and I won't be able to go through it again. And I put this up there only because I figured out in PowerPoint that I could put up the text that is transparent. And I thought that was a pretty cool thing. And so I decided I wanted to use it up there, and that's why I put that there. And so we're trying to look at the applied ecology of phage therapy at this point. And so people are trying to use phages to treat bacterial biofilms. We'll get into more of what that is in just a sec. And it's my feeling that phages, when they're exploiting biofilms in the wild in order to make more phages, do so in a way that's not terribly helpful for actually eliminating biofilms from the environment. And in fact, we can help these phages in a number of different ways. And therefore, we suddenly have an applied ecology rather than an ecology. In fact, we can attempt to mechanically remove biofilms before we apply the phages. And this might reduce the number of bacteria that are targeted, but also increase the ability of the phages to get to the bacteria. And there are actually a number of different organisms that consume biofilms by mechanical removal of the biofilms. But in terms of medical considerations, we also have debridement, where we take off basically the necrotic tissue and the pus and what have you. And this probably increases the ability of the phages to get to the bacteria. And they in fact use that when they're treating infections that look like wound infections. We can actually use enzymes that are associated with phages to dissolve the extracellular matrix that makes up these biofilms that potentially can allow the phages to reach the bacteria much better. But this might not be helpful to a phage by basically dissolving a biofilm in a wild context, because the phages probably want these bacteria to be near each other so they can more easily go from bacterium to bacterium. There's also phage enzymes, the endolysins, that we can use to kill the bacteria that perhaps make up the biofilms without, in fact, making any phages at all. But really, if you're going to try to treat a biofilm, rather than just adding a few phages and hoping that they'll take over the biofilm and get rid of it, what you want to do is add a fairly substantial number of phages, and then add them again, and add them again, and add them again over perhaps multiple weeks or even months until you've depleted the infection to the point where the body's immune system can take over. And so ecology of phage biofilm interactions is the next obvious thing. So bacteria can live in spatially structured environments. Spatial structure simply means limitations in movement. It could be bacteria that in fact are tied to one another in cellular arrangements. They could form microcolonies that in fact can make up biofilms. Spatial structure is important in the development of phage plaques, which are clearings of bacterial lawns that are associated with phages that are not able to move very far from where they were born from an infection, and then lots of other things. And so first considering microcolonies. So microcolonies make up many biofilms, and we can consider the ecology of biofilms from the perspective of microcolonies. If you have a bacteria that comes along and it initiates a microcolony, it basically attaches to a surface and starts replicating. At a steady state, so these things, the job of these things is to make more bacteria that'll found more microcolonies. At a steady state, no matter how many bacteria are going to be released from this microcolony, you're only going to have, on average, these bacteria founding one new microcolony. So you can have a population that's growing, so you can found more microcolonies. You can have a population that's declining, so you found fewer microcolonies. But at steady state, where nothing is in fact changing, one bacterium is going to found a microcolony on average. It's going to produce, no matter how many bacteria it produces, most are not going to found microcolonies, on average only one will. And so we can consider that same idea for phages, where rather than a microcolony, you have something that's equivalent to a phage plaque, where you have a bacterium that finds its way into a biofilm-like situation. It replicates some more within the same location. You can call it a focus of infection. And you would have the same expectation that within this focus of infection, no matter how many phages are produced and released, at steady state, on average, there's only going to be one phage that survives to found a new focus of infection. So these phages are going to make lots of babies, potentially, but it's a harsh environment out there. And in fact, there probably isn't a whole high lot of density of biofilm out there to attack. So they're going to spend a lot of time searching out for a new biofilm to initiate infection in, and most of them are going to die trying. And that leads to a theoretical, I guess, model for what phage exploiting biofilms is all about, where you've got what I call explorer phages that are just diffusing out there outside of biofilms, and by chance they might collide with a biofilm, collide with a clump, a microcolony of bacteria, infect the bacterium, go through their infection, lyse the bacterium, release the phages, and infect more bacteria of that same clump. Or maybe there'll be adjacent microcolonies of the same bacterial type. They'll be able to diffuse to those microcolonies within the biofilm and initiate the same process there. Or maybe there'll be more distant microcolonies within the same biofilm that they can diffuse to. And together within here, they are generating phages. And some of those phages, unlike within a plaque on a plate, a petri dish, some of those phages are going to in fact diffuse out of the biofilm. And those are going to be the phages that go on to found new foci of infection. So basically, these phages up here are these phages there, and this are kind of a representation of the same thing. At steady state, no matter how many phages these guys are making within these foci of infection, on average only one is going to survive to initiate a new focus of infection. And so these guys, it might seem like a cushy life. They found a place to infect and they've got lots of new infections they could possibly initiate, but it's a harsh life out there. And when they leave the biofilm, there's not a whole high density of biofilms within, say, an aquatic environment. And so it'll take time until they find a new biofilm where they can successfully infect. And in the meantime, a lot of the phages will become inactivated. And again, you can have a population where the phages are growing in number, in which case you'll have more than one survive to initiate a new focus, or you can have ones that are declining. But at steady state, on average, only one is going to survive. And then this is my stuff where I look at the ecology of biofilm and phage interactions, and also phage ecology within plaques. And so we can talk about 4 different kinds of treatments. The passive treatment is inundative treatment. You need bacterial cytal activity, but not necessarily lysis or amplification of the phages. Active treatment, you need amplification. Passive active, you need to kill the bacteria, and that might be enough, but you've got this bonus that you're making more phages. And then with what I call active penetration, the lysis of the cells, and maybe the amplification, is contributing to removing the bacteria layer by layer in clumps in biofilms and maybe getting rid of the bacterial infection. So you might have to do your dosing again and again to be successful in that active penetration. And then finally, that's not what I do for a living. What I do is actually teach way, way too much. And I turn my teaching into a website. And the website's huge at this point, as you probably would expect, given everything else I've shown so far. And right now I've got about 250,000 people per year that use it, with about 500,000 page views. And I actually have 2 online textbooks of sorts that are on that site. And it is not yet 4 o'clock, and that's it. Thank you very much.
We have maybe time for exactly one short question, but of course, if you can meet up with them later or send them an email or something if you have additional questions, go ahead.
Is a similar thing thought about for fungal infections? Are there viruses or phages that left for fungal?
It's an excellent question. Well, they're not phages, they're fungal viruses or mycoviruses. And in fact, people do attempt to use mycoviruses as antifungal agents. One of the problems is that fungal viruses are very weird. And so you don't get this, a virus goes in and it lyses the cell kind of thing. And so it changes the quality, the characteristics of the treatment. But people actually have used viruses in order to reduce fungal infections. I believe it is chestnut blight where that's been most successful. We actually use viral infected strain of fungus of the same species to reduce the actual pathogenic strain. And in theory, I'm actually writing a chapter on this, not because I want to, but because I'm editing a book and somebody's got to write the chapter. So I'm actually including not just phage therapy, but antifungal therapy and anti-protist therapy, which is completely hypothetical, but it could be. There are 2 papers that have been written on that so far. And also anti-archaea, where you can potentially use phages to treat methanogens, for example, in a bovine rumen, in order to reduce the amount of methane that's produced. It's somewhat hypothetical as well, but it's more progressive than this. But yeah, excellent question.
Great, well, thank you, Steve, that was awesome.
Yes, I'm available for questions. And